Now that you've completed the miniPCR Lemur Lab, it's time to interpret your resulting gel. This video will explain how to use the information in your gel to determine each lemur species. Remember that in this lab, we used PCR to copy a 1200 base pair section of the site B gene. Then we added a restriction enzyme that cuts site B at the site of a mutation carried only by the furry-eared dwarf lemur. The restriction enzyme cuts the 1200 base pair PCR product into two fragments, a 500 base pair fragment and a 700 base pair fragment. This means that when you see 500 and 700 base pair bands in a sample, you can infer that the DNA came from a furry-eared dwarf lemur. If you see a 1200 base pair band, you know the DNA came from a lemur belonging to a different species. You used gel electrophoresis to analyze the experimental results. Now, looking at the gel, we'll use the DNA ladder like a molecular ruler to determine the lengths of the DNA fragments in our samples. We'll focus on these bands in the ladder, which contain DNA fragments 1200, 700, and 500 base pairs long. The DNA fragments we expect from our experiment fall into this range. In the first sample, we see two bands. This band contains DNA fragments 500 base pairs long, which we know because it traveled about as far as the 500 base pair band in the ladder. This band contains DNA fragments 700 base pairs long, which we know because it traveled about as far as the 700 base pair band in the ladder. This banding pattern means that this sample contains DNA that was cut by the restriction enzyme, so we can conclude that it came from a furry-eared dwarf lemur. In the second sample, we see one band. This band contains DNA fragments 1200 base pairs long, which we know because it traveled about as far as the 1200 base pair band in the ladder. This banding pattern means that this sample contains DNA that was not cut by the restriction enzyme, so we can conclude that it did not come from a furry-eared dwarf lemur. Now you can compare the data from our experiment to these results to identify which lemurs are furry-eared dwarf lemurs, and which, if any, belong to another species.